Hello everyone. We analyze high-C interaction networks and focus strictly on topological properties of these networks. The main questions we are trying to answer are: can topological properties of interaction networks distinguish between different cell types? And also, what are the most important of such properties? And secondly, what is a typical structure of interaction networks? And can we assign a biological significance to some structural elements or features? DNA is a linear strand, but it is folded many times, forming a complicated three-dimensional structure. Because of DNA strand folds, some loci are located close to each other. High-C technique captures information about closely interacting genomic regions at a genome-wide scale. We study high-C data as graphs, where DNA segments are vertices and the captured interactions are the edges. Here is an example of a network component that we study. For each of the datasets in our study, we have a separate interaction graph for each of the chromosomes. Currently, we have three high-C datasets that we have studied. First, the promoter-captured PC high-C dataset of 17 blood cell types obtained from a single experiment. Second, the PC high-C dataset of 20 different human tissues, such as lung, aorta, and liver. Third, the high-C dataset of 10 human tissues, where the covered set of tissues is a subset of those that are included in dataset 2. In our study, we assess how well various network topological features for the networks are able to discriminate between various cell or tissue types. For dataset 1, quantitative distances between 17 cell types can be derived from a hematopoietic tree that has been published as part of supplementary materials for the PC-High-C experiment. For analysis, we have used continuous distances as well as several discrete threshold or tree topology-based distances. For datasets 2 and 3, we have used distance matrices derived from RNA sequencing data by calculating transcriptomic signature distance as proposed by Manatakis and colleagues. We characterize topological features of graph by topology metrics, generally using all graph properties that can be efficiently computed and are present in available networks. The initial set of metrics contained 57 such properties, and these perform even surprisingly well for discrimination between different cell or tissue types. For example, for dataset 1 and continuous tree-based distances, we obtain a correlation of 0.82 between metrics and tissue distances. Notably, it turns out that the set of metrics can be narrowed down to just 6 metrics without losing precision. The metrics are related, with some of them being quite similar, as we can see in the picture. In the end, we have selected the metrics that we can assign some presumptive biological explanation, such as number of cliques of size 3, number of edges in the largest strongly connected component, sizes of strongly connected or biconnected components, number of cycles of length 2. When comparing certain metrics with tissue relatedness in our data, we observe occasional positive or negative correlations, such as in this case, where cliques of size 3 are slightly more prevalent in closely related tissue types than distantly related tissue types. An interesting side effect we discovered but couldn't really explain. One might expect that our metrics would perform similarly in all chromosomes. However, if we look at how well our metrics perform in different chromosomes, we can see that chromosomes cluster into groups. There are visible correlations between certain metrics and tissue relatedness, especially in the blood cell PC-High-C dataset 1. We have also used the same methods on datasets 2 and 3, yet those two datasets are less dense, less stable across different chromosomes, resulting in lower metric correlations. It is, however, still possible to choose metrics and create models to distinguish tissue pairs from each other and from other datasets as well. Another notable topological feature is the structure of connected components in networks. We discovered that we can find a rather large set of tissue types that all have most of the links of the connected component, while there is also a set of other tissues that contain very few of the links of a component. This way we can find tissue-specific components. This component structure has been quite extensively studied for dataset 1, and we have developed an algorithm for automated detection of potentially interesting components. Similar component structure was also discovered for datasets 2 and 3. The component properties, however, are slightly different, which is not surprising since the networks themselves describe different types of biological features, and these datasets have not yet been extensively analyzed. So, all in all, metrics that count graph elements in overlapping subgraphs are useful in quantifying the topology of high-C graphs. Topology is different in different datasets and in different chromosomes, and more study is needed to find a set of metrics that would distinguish different cell types. See you in medicine.